Hi biologists, let's start by looking at the learning objectives for this lesson. At the end of this lesson and following the biology syllabus, you should be able to give a detailed account with the aid of diagrams of the stages of mitosis. What does this actually mean? What are we trying to learn? Well, it's straightforward enough. You have to be able to describe in detail, with the help of diagrams, the process of mitosis. Let's look at a detailed study of mitosis. You might remember the cell cycle from a previous video. The cell cycle, or the life cycle of a cell, can be divided into stages. Interphase is the stage in the cell cycle during which cells are not dividing. Mitosis is the division of the nucleus to produce two genetically identical daughter cells. Because this is a detailed study, you must also understand that mitosis is usually followed by cytokinesis or cell division. This is where the cell or the cytoplasm divides to form new cells. Now, strictly speaking, the objective of this video is to have a detailed understanding of the process of mitosis or the process of nuclear division. An understanding of the cell cycle stages helps the understanding of mitosis. So, a detailed study of mitosis involves watching the following videos in the sequential order given. Mitosis number 1, which describes the process of interphase. Mitosis number 2, which involves a detailed study of mitosis, and mitosis number 3, which describes the process of cytokinesis. Let's look at cytokinesis, or division of the cytoplasm. Reminding ourselves of the definition of mitosis, we can see that mitosis is nuclear division, in which one nucleus divides to form two new nuclei, each containing the same number of chromosomes with identical genes, leading to the formation, really, at the end of the day, of two new genetically identical cells with the same number of chromosomes as the parent cell. So, we can see, following mitosis, that it results in the formation of two identical daughter nuclei, or two new nuclei. The process could then stop there. We see that in the formation of the pollen grain, whereby the pollen grain nucleus divides by mitosis, leading to the formation of two new nuclei, the tube nucleus and the generative nucleus. However, under normal circumstances, mitosis, division of the nucleus, is followed by cytokinesis, or division of the cytoplasm, leading to the formation of the two genetically identical cells. After the nucleus has divided, therefore, the cell will divide in two, or the cytoplasm will divide. However, the process is different between animal cells and plant cells. Don't forget, cytokinesis, strictly speaking, like interphase, is not strictly part of mitosis. How does cytokinesis happen in animal cells? Well, in animal cells, the cell membrane pinches in. A cleavage furrow is formed. The cleavage furrow will deepen and eventually divide the cytoplasm in two, leading to the formation of two identical cells, each with the same number of chromosomes as each other and as the parent cell. So, due to cytokinesis or cell division, the cell basically splits in two. In plant cells, the process is slightly different. There is no cleavage furrow or splitting or pinching in of cytoplasm. This is because the cell wall is too strong or too rigid, so the process goes a little bit differently. Small cellulose-carrying vesicles line up across the cell, forming a cell plate. This cell plate will form new cell walls for the neighbouring cells. The region between the walls is called the middle lamella, resulting in the formation of two new identical cells with the same number of chromosomes as each other and as the parent cell. So, in summary, when we look at cell continuity, which is the ability of cells to divide and survive from one generation to the next, we can look at it under two concepts or two headings. We can look at it from the point of view of the cell cycle, which are the changes that occur between one division and the next, or we can look at this cell division totally from the point of view of nuclear division or mitosis. If we look at the changes that occur between one division and the next, if we look at the cell cycle, and if we have to describe the cell cycle, we have to include all the phases. We have to include an account of interphase, where the cell is not dividing, mitosis, which involves nuclear division, subdivided into 4 stages, prophase, metaphase, anaphase and telophase, and finish the account with cytokinesis or cell division. We can also just look at it from the point of view of mitosis, where basically we're just concerned with division of the nucleus. You basically take out the account of mitosis out of the cell cycle and just concentrate on that nuclear division alone. That's why we've said in earlier videos that, strictly speaking, interphase and cytokinesis are not part of mitosis or nuclear division. Now, how do we remember these phases? Because they can be rather confusing. So I've come up with a little memory trick. To remember the phases of the cell cycle, we just remember this. I paid Michael a tenner change, obviously helping us remember the phases of the cell cycle. I in interphase and I for I. P for prophase, P in paid, and so on. So I paid Michael a tenner change gives the full account of the cell cycle, including the important process of nuclear division. Well, if we just want to describe mitosis on its own, which is really the objective of this lesson, or previous lessons, well, we just give an account of the 4 phases. You basically just paid Michael a tenner, or PMAT, as we suggested in a previous video. Hopefully this makes mitosis a little clearer and that we're not in two minds about it. There you have it. Practice in a jotter. Now that we have reached the end of our lesson, have we achieved our objective? Can you give a detailed account with the aid of diagrams of the stages of mitosis?